My name is Eileen Sullivan. I'm from the Boston area, and I have had the privilege of knowing Zach Hunter since before he was Dr. Hunter. I've been treated at Dana-Farber for decades, and he has also been very supportive of our support groups that meet at Dana-Farber. So I think it's possible that at some meeting or another in the Boston area, somebody introduced him and said, I would like to introduce for the first time Dr. Zachary Hunter, because he had finally done his PhD work. And he was just reminding me or telling me that he switched from math to science, having worked with Dr. Treon at Dana-Farber. And what a good decision that has turned out to be for all of us. So Dr. Hunter is now, of course, Dr. Hunter. And I used to introduce him back in the day as a wunderkind. But given the gray hair, the kids, all of that, it feels a little odd. So he may not be a kind anymore, but he's certainly still a wunder, because he has a remarkable mind for thinking about Waldenstrom's, asking the right questions, finding the answers to questions people haven't even thought of yet. And so he's the, let's see, what is his official title? Associate Director of the Bing Center for Waldenstrom's macroglobulinemia at the Dana-Farber Cancer Center in Boston. And we are just so lucky to have him. If this is your first time hearing him, be prepared, because you might want to take some notes, or you might just want to listen and take it all in. He has a gift for explaining this stuff in a way that non-scientists can handle. And I have heard him many, many times. I learn something new each time, not only because I'm learning more each time, but he's learning more each time. And so there's always something new on the horizon. And fortunately for me, I heard a lot of this talk a few weeks ago because he graciously came and met with our support group in Boston for our first in-person at Dana-Farber support group meeting since before COVID. So we had a good crowd, and there were lots of questions, so I'm sure that will be the case again today. I also wanted to mention his folks are here today. And so I believe we have them to thank for, for Dr. Hunter. So would you mind just standing up and giving a wave so everybody can realize the source of all this knowledge? All right. And I know there's more things to say, but they're in the program, so you can read about his accomplishments. Oh, but just one thing. Has anybody ever heard of the MYD88 mutation? Raise your hand. Raise your hand if anybody's heard of that. Okay, guess who found it? Guess who found it? He likes to say, I didn't really find it, but who was on the team that found it, and who probably asked just the right question in order to get them that information. So I'd like to present to you Dr. Zachary Hunter from the Bing Center.
Well, thank you so much for that introduction. It's really great to be here. So, yeah, we're going to be talking about the science of Waldenstroms. And for those of you who have not heard me speak before, really, please don't worry. We're not assuming a great deal of background knowledge. If you're thinking back and be like, what happened in high school biology? Honestly, I don't remember either. So it's okay. And so we're going to build things up slowly. We're going to focus on big concepts. So I am going to talk about some genes and some pathways, and I'm going to tell you right now, none of it's important. Those names are not important. If there's an exception to that, I'll make a note of it. But for the most part, really try to focus on the big picture ideas. So as Eileen said, my name is Zachary Hunter. I work at the Bing Center for Waldenstrom's macroglobulinemia. I'm the associate director of the program, and I run all of the genomics and large data analysis efforts there at the center. And for those of you who have heard me speak in the past, I often focus on the role of DNA and how DNA gets changed in cancer, and in Waldenstrom's what that means in terms of how we're directing our new therapeutics, how people respond to therapeutics. I'm not giving that talk today, just so you know. This is a new talk, new ideas. Actually, some of this stuff is really cutting edge stuff that has only really been presented at the most recent international conferences. So really wanting to let everyone here have a sense of some of the excitement and some of the real breakthroughs that are happening here in this disease. Now, this is a great opportunity to mention, however, that those older talks are available online. The IWMF has an amazing website and a great archive of talks that have been given at the Ed Forum, not just by me, but really top people in Waldenstrom's from going back a number of years. They're on YouTube, again, hosted by the IWMF. Really check them out. There's some wonderful stuff there. So just putting a little plug for that if you get a chance. So I want to start off by talking about DNA. DNA is actually really simple. It's easy. Genomics is super straightforward. Just trust me on this, okay? So DNA is made up of 4 different things called bases. We abbreviate them A, T, C, and G. And that's it, you're done. Okay, we can move on. No, but actually there's a special trick, which is that they're complementary. A is pairs with T's, C is paired with G's. And this means that you can create 2 parallel strands that are perfectly complementary to each other. Now, this is not just very stable, right? And this is why, for instance, you can go and dig up old mummies and find out what genetic diseases they had. But it also has a lot of advantages. You can pull it apart and then just read one side. If something gets damaged, right, you can look at the opposite base. You can say, oh, hey, there's a T on the opposite side. That damaged base, that should have been an A. Let's repair it. Beautiful structure, beautiful engineering, right? So you have about 3 billion of these. It's a lot. And they're organized into 23 long strands. And they're called chromosomes. Have you ever heard of like 23andMe? The 23 they're referring to are these chromosomes, these long strands of bases. And that really is it. That is genetics, right there. Everything else is just details. So another concept that you may have heard of are genes, right? What is a gene? You associate genes with DNA, but how? You only have, well, you technically have 2 copies, and we'll get to that, of your DNA everywhere. But you may want to use a particular section of your DNA more than others, right? So you don't want to actually send your DNA out to do things. You just want to copy little chunks of it out, right? So these copies, these little mini copies, are single-stranded copies called RNA. And anything that gets copied off the DNA and sent out, right, that's a gene. Congratulations. Again, that's it. That's the big concept. That's the secret. Now, some of you might feel a little bit unsatisfied. That's okay. I want to encourage this, actually. This satisfaction is your inner scientist that all of us have, saying, that's great, but there's something more here, right? This doesn't quite hang together right. So let's think about, these are letters. We have 4 different letters. How do we use them? What do we use letters for, right? I mean, just think about it in our lives. We use letters to do what? Make words, right? What do we use words for? Make senses and messages, which we then send out into the world, right? DNA is no different, right? These copies that are being made have words. Groups of 3 of these bases actually encode for a particular amino acid. Now, you've probably heard about that in like nutrition and stuff like that. Oh, you got to get your right balance of amino acids. Well, this is because all proteins in your body are actually just long chains of amino acids. So what are these messages? These are instructions on how to build these chains of amino acids or how to build these proteins, right? So it's very, very similar to the words that we use, just, a little bit more basic. Now, okay, if we could summarize all of genetics, right, in just a few minutes, why do we study it? I mean, it seems like, we could just, your PhD one and done, I could just mint all of you guys, and you'd be good to go. It Turns out, if you take a bunch of really simple things, say 3 billion of them, and you put them together, you can get some very complicated emergent behavior. And so a great example of this is, I'm sure everyone's heard of things like ChatGPT, right? Computer code is actually just made up of ones and zeros, right? We have 4 bases, they only have 2, right? Put enough of those ones and zeros together, and they have a lot less information, right? Theirs are literally one big strand. Ours, we didn't talk about it yet, but there's 3-dimensional modeling and where the genes actually are in the cell and all the stuff that you can do for encoding additional information. They only have just long strings of ones and zeros, and that's it, right? And yet, they can beat us at Jeopardy, right? So think about that. Very simple things like true and false, you get enough of them together, you can make ChatGPT, you can make IBM's Watson. So we're no different, right? Just when you have a lot of simple things together, you get very complicated behavior. So let's just review, because I said it's simple, but it is still a lot. We have DNA, right? 4 bases, sections of it copied into RNA, those are genes. What do genes do? Genes encode for protein. Protein does all the work in the cell, interacts with the environment. All right? That's the basis of modern molecular biology. So how does this play out in WM? How does it play out in cancer? Well, mistakes happen. They do, at least for me. But it happens in our DNA too. Sometimes DNA gets damaged, and despite this wonderful structure of complementarity, it isn't repaired correctly. And if these are encoding for messages, these genes, if you change the code, you change the message. And if you do this enough, genes are going to start to lose some of their function or gain new ones. And when enough of that happens, cells can start to grow out of control. And when that happens, we call it cancer. And so the most famous mutation in Waldenstrom's is in this gene called MYD88. Now this is one of these places where I'm going to say, you know what? There's a lot going on in this slide. Don't worry about it. The only thing I want you to look at right now is the MYD88, the 2 of them coming together. Right? That's the only thing that's important. And trust me on this. Trust me. You're not going to want to look at the rest of this. It's a waste of time. MYD88 is actually part of your innate immune system. I like to think of it as your dumb immune system. Right? You have an acquired immune system, very smart. It's cool. It's why vaccines work. It's why you don't get the same disease over and over again. The problem is it takes time to kick in. If you waited for it to kick in, you'd be dead, honestly, right? Really, it's powerful, just slow. So you have this early warning system that's in all of your cells that is good at seeing things that are common in bacteria and viruses, right? So they shouldn't be in your body. It doesn't know anything else. It's just like, hey, bacteria, virus, let's freak out. Let's fight whatever's there. We don't have any details. And so when that happens, that will allow 2 different molecules of MYD88, this protein, to come together. And by doing so, it allows other proteins to allow the form around it to form a large signaling complex. And so what do you do if you see some bacteria or virus in your environment? Clearly not supposed to be there, right? You don't sit around, right? You try to fight it, you try to survive, especially if you're an immune cell, you try to grow, and you try to recruit other cells to come in and help you, right? And so how is this done? Well, there's this big signaling cascade, which I just told you to ignore, and you should continue to ignore, and I'll tell you why in a minute. But it will eventually result in this protein that's actually held out, sort of in the exterior part of the cell, inside the cell, but sort of on the periphery. It'll cause it to be released, where it can then go and start interacting with the DNA. And it's these DNA binding proteins that actually help control when genes get used. So when it starts to interact with the DNA, this inflammatory signal gets generated that helps the cell grow and survive. And we can see it, right? Turns out we have tools that we can use in the lab, unfortunately not in patients. You put it into patients, human blood will just destroy it immediately. But in test tube, we can do this. We can prevent mediate from coming together artificially. And so one thing we can do is we can color the area in a cell. Looking at these pictures over here, we can color the area where the DNA is in blue. It's called the nucleus. We can color the other parts in red. And then we can label this DNA binding protein that's downstream of MYD88 that's activated in this sort of green color. And you can see these are a cell line, actually, of Waldenstrom cells called BCWM1. And when they're resting, all that activated NF-kappa B is there in the nucleus binding to the DNA. If we put an inhibitor in, it all leaves. You can see over here, right? It's no longer in that blue area. So you can start to get the sense of how a mutation in the DNA and how signaling goes back and interacts with the DNA and back and forth. Now, why does this matter? This mutation in MYD88, what it does is it's a gain of function. It's the same mutation over and over and over again in the vast majority of patients. And what it does is it allows these two copies to come together regardless if these receptors are active. You no longer need to see a bacteria and virus. They can come together by themselves. Therefore, it always sees a bacteria. It always sees a virus. It's always signaling. It's always asking for help. It's always trying to grow. It's always trying to survive. Sound familiar, right? Now, why did I tell you to ignore all this? Well, that's because we had a wonderful, wonderful guy working who used to work in our group named Guang Yang. And he made a critical insight. And that was that, well, this is the canonical pathway by which this works. In Waldenstrom, is actually not the main way it signals. The vast majority of this signal goes through a molecule or a protein that we call Bruton's tyrosine kinase, or BTK. Some of you may have heard of this, right? This is the target of ibrutinib, of zanubrutinib, pirtobrutinib, acalabrutinib. All these are targeting BTK. Why are we targeting BTK in Waldenstrom's? It's because it's directly downstream of this mutated MYD88. We're trying to block that signal, right? That is why we're doing this. So another example of a mutation in Waldenstrom's is CXCR4. And in CXCR4, it's a sort of good counterexample to MYD88. We see it in about 30 to 40% of patients. And what's interesting here is it mediates the same thing over and over again, leading to a new function, right? The ability to bind in the absence of a signal. Here, we're losing a function. We're losing the ability to reset the receptor. All these different mutations all occur right here at the end, doesn't affect signaling, but blocks the ability for the receptor to reset. You might think, well, okay, so it doesn't work anymore. This is where things get interesting. If you block the ability for something to reset, that also means that once it's turned on, you can't turn it off again. And so what happens is this gets turned on, loses the ability to turn itself off, and will sit there and signal forever. Now, what does CXCR4 do? It's a group of proteins that collectively are called chemotactic receptors. It's a fancy way of saying it helps cells follow a chemical trail to a particular location, helps them migrate through the body. And in the case of CXCR4, where it's trying to migrate to is the bone marrow. Again, that might sound familiar. Interestingly, and maybe accordingly, we see a lot less, for instance, people with involvement in their lymph nodes if they have CXCR4 mutations. And again, we believe this is because those cells are now really focused on homing to the bone marrow instead of to other parts of your body. All right, so we've talked about mutations in cancer, but there should still be this nagging feeling in the back of your head, right? I'm trying to paint this picture of how cells work, and something should feel incomplete. Again, listen to that, right? Because we're going to keep digging. So the first thing that should occur to you, or might occur to you, is, how big is 3 billion base pairs, right? I mean, 3 billion is kind of a big number. Well, it turns out it's really big, right? If you were to stretch these things end to end, you're talking about 40 inches. And what we don't usually tell you is you don't have one copy of that. You have 2 copies, right? You have one copy from your mom and one copy from your dad, right? And that's inside every single cell in your body. I mean, that's impressive, right? That's an impressive amount of information in such a small space. It's not just in the cell, it's in a subcompartment of the cell called the nucleus. So how does this work? Understanding how this works actually gives us a lot of insight, both into cancer and to actually how cells operate in general. And that's because the DNA gets wound up really, really tight. So tight that actually some genes simply can't be seen. So you can control which genes the cells are able to use and see by whether they're wound or unwound. And this gives us a novel way of controlling how cells respond to their environment. There's additional things we can do as well, which is we can chemically annotate the DNA, and the cells do this. And this can, we talked a little bit about DNA binding proteins. These chemical changes can either inhibit or enhance the ability of these binding proteins to interact with the DNA. So you can annotate it in a way that will encourage this DNA binding or discourage it, right? Effectively silencing a gene. So this now is giving us a slightly better picture of what's going on, right? Because if you thought about it in advance, like, well, hey, this DNA, RNA, protein thing is great, but how does the DNA know what to do, right? You need some kind of feedback mechanism, some kind of control or response, right? Or none of this really makes sense. And that's the epigenetics, right? That's this wrapping and unwinding, this chemical annotation. So what happens is proteins get made, they interact with the environment, they bind to the DNA, activating new genes, but also changing the structure, wrapping them and unwrapping them, allowing different genes to be visible, allowing then different patterns for transcription in different proteins to respond to that environmental stimulus, right? And then back and forth and back and forth. And now you have this dynamic system. You can actually have something that can respond to new situations dynamically instead of statically. And we collectively call these the genome, the transcriptome, that's looking at how genes are being used, the proteome, looking at the status of proteins in the cell, and the epigenome, which is looking at all these chemical annotations and the structural changes, this sort of wrapping and unwinding of the DNA, allowing which genes can be seen and not seen. And to give you an example of how this works, right? So you have this example here of this sort of nice loop of protein, DNA, RNA, sort of all interacting to each other in response to the environment. Is I want you to think about your computers for a minute, right? Because there's this sense that a lot of people have that like DNA is destiny. And I want to push back on that, and I want to say DNA are tools. Okay? And what do I mean by that? Everyone here probably has a laptop or a tablet or something, smartphone. But in reality, it's probably a Mac or a PC. Yeah, I see the one person who's a Linux user out there, and I'm with you. But for the most part, Mac or PC, right? And all of your computers, your operating systems are all clones of one another. They're perfectly identical. And your operating system defines all the tools your computer is allowed to do, all of them, right? So why are all your computers different from each other? You've installed different applications on them. You have different family photos on them. It's the interaction of its environment with those tools that allows those computers to be different, right? What's the environment? It's you, right? So how does this play out with people? Actually, very similarly. We look at twin studies, and popularly we always talk about the way that twins are all the same. They use the same toothpaste, date people with the same names. But actually what's really more fascinating than that, if you meet identical twins when they're really young, they're really similar. I mean, it's really wild. But when they're older, they're actually quite different from each other, right? And this is because they were born with the same set of tools, right? But they responded to their environment differently. They had different experiences. They made different life choices, right? And that interaction caused them to evolve in different ways. So it's actually more common that the identical twins wind up being quite different from each other than not. And that's the reason. So DNA is not destiny. It's just the set of tools that your body has to respond to its environment. Okay, well, that's a nice story. But in the back of your head, that inner scientist should still be unsatisfied, right? Because let's think about this. That same DNA is in every single cell in your body. So why do stem cells look and act differently from the neurons in your brain, differently from the fat cells, different from your blood? They don't look anything the same. They don't act the same. How is that possible? Same DNA, right? Well, we can say, okay, they have different proteins, right? How are those different proteins controlled? Well, we just discussed this, right? Transcriptional changes, how genes are used, right? There's RNA. Okay, cool. Well, why do they have different RNA patterns? Right? Their epigenomes are different. They're controlled differently. So I've been focusing on how rapid changes, how cells respond to their environment. But in reality, a lot of these epigenomic changes are actually designed to be more or less permanent, sealing off such sections of the DNA that will then never be used by that cell. This means that different cell types effectively have their own unique genomes based on which genes they're allowed to see. Kind of clever, right? Solve two problems at once. How do you make it all fit? How do you keep them all different from each other? Get these unique functions in different cell types. So by controlling which genes you're allowed to see, you get these different types of cells. Now, I imagine some of you can think about where I'm going with this. We talked about mutations in cancer, how you change the message and therefore change the function of a gene, right? Well, what if you were to change the genes that the cell's allowed to see? You could start getting genes they're not supposed to see added to the mix. Genes that are supposed to be there, supposed to keep them in check, might suddenly disappear, right? No mutations involved. Changes in their epigenome, right? This is a critical part of cancer development, right? Now, we've gone through a lot of pretty heady stuff, and this is it. It's all downhill from here. So I want to talk about stem cells, right? Now, classically, when people think about stem cells, they think about, okay, cloning people and being able to make every single cell in your body. That's true. There are stem cells like that from embryos. That's not what most stem cells are. Most of them are specialized. They know how to make your skin, for instance. When you get a cut and it heals, thank your stem cells, right? Your blood gets replaced all the time, right? There's a stem cell that knows how to make every single different part of your blood, your immune cells, your red blood cells, everything, right? And it's constantly working to make sure that all those get made and replaced all the time, right? And how do they do that? They do it by controlling their epigenomic states, okay? And so this is where this interaction with cancer gets really, really interesting. So what if you have an identical set of mutations occurring on different stages of development, right? They effectively have different sets of genes that they're able to see. The impact of those mutations are going to be slightly different, right? And so the theory here is that you can wind up with different subtypes of a disease by basically having the disease start in slightly different places in development because they're seeing slightly different genes. And this is the cell origin model of cancer. So how does this play out in WM? And this is where we start moving into some of the more sort of cutting edge stuff. So some of this may change over time, but this is sort of our current understanding. So this is, we talked about the chemical annotations on DNA. This is that, right here. So what is this graph? This is called, what I'm going to start referring to as fancy math. The details don't matter. So what we're doing is we're taking all this information about the chemical annotations and we're trying to put samples that are similar to each other, close to each other on the graph, and samples that are very different from each other, very far away, right? And then plotting all out and sort of seeing where it lands. That's all you really need to understand about this. But what do we see? We see two distinct clusters, right? One cluster has a lot of orange dots. These are ones who have CXCR4 mutations, and a few purple dots. These are the ones who have just mediate, no CXCR4. And they're clustering around a lot of blue dots. And what are those? Those are actually healthy donor memory B cells. This is the cell type that we've long thought that Waldenstroms might develop from. So that's kind of interesting, right? Because these epigenomic states are fairly durable. And the reason for this is that if you can imagine, I'm working on a house renovation right now, and let's say I wanted to change out the windows of the house. I'm not going to tear down the house. And that's crazy, right? You just take out the windows, right? You put in new windows. Easy, right? Cells are the same way. They need to evolve around some aspect of their epigenomic limitations. They'll change those things, right? They're not going to destroy everything. And so you're left with this fingerprint of what they once were, because basically the cells are too late to get around to changing it. Very, very thoughtful of the cells, really. So what's this other group over here? Well, they're not clustering with these healthy donor memory B cells. They're clustering actually with IgM multiple myeloma. Now, this is a completely different kind of disease. They are unlike multiple myeloma in every single way. I don't want anyone to get concerned or confused on this point, except for the fact that it does look like their origin may be similar. All right? So multiple myeloma is a plasma cell disease. Memory B cells are B cells. So let's take these two groups and let's just give them a name, just for kicks. Right? So we're going to call this B cell-like Waldenstroms and plasma cell-like Waldenstroms. And let's just remember that as we move forward and keep talking about some of these findings. So as a bunch of you know, we've been working on this large study that we called the 300 Project of trying to collect data on 300 untreated Waldenstrom's patients and trying to understand their genomics, their transcriptomics, and their epigenome, right? And really make a model of what is happening in Waldenstrom's and use this information to develop new therapies and better treatment, right? And one of the things I wanted to look at when we first started doing this is a signal we had associated with CXCR4. And before we go there, I just want to quickly take a look at these graphs, because I fully realize that many of you aren't used to looking at box plots, right? That's what these are. They're really easy to understand. Each dot is a patient. The higher up the dot is, the more gene expression we're talking about. These are all looking at different expressions of different genes. This big, thick line here in the middle, it's called the median. It means half the data is above that line, half the data is below that line. The box, that's 50% of the data, the inner 50%. That means 25% of your data is above the box, 25% is below the box. Why this is useful, it means you can just, instead of trying to look at the dots and where they all land up, you can just kind of look at that and immediately be like, okay, 50% of the data is right here, here are kind of the extremes. We can compare these two groups, get a sense of how they're different. So that's all that's happening here. It's not takes a little getting used to, but it's all right. But this is what bothered me. If you took patients who had MYD88 mutations, but not CXCR4, we had this very strong gene signature, really weird genes being expressed. But then you added CXCR4, you had MYD88 with CXCR4, all those genes went away again. And that really bothered me. Why bother to go through all this work to get these genes expressed only to take them back, right? That feels counterintuitive, right? But that wasn't the only thing. The other thing that bothered me were these samples. There were groups of patients who had CXCR4 mutations but didn't have the signature. And as we started expanding it more, there were a bunch of people who didn't have CXCR4 mutations but did have the CXCR4 signature. And this made me start thinking, maybe we're wrong. Maybe this isn't CXCR4. Maybe there's something else happening here. Maybe there's a background that CXCR4 likes to occur on. And that's what we're really measuring here. So we went out to try to find it. And this is what we got, right? We wound up with 3 distinct groups. So red here means strong expression, blue here means less expression. These are all different genes kind of being aggregated together. But you can just look at it, 3 distinct groups. And you might recognize some of these names. because these samples overlap. We can match them to each other. These are, in fact, the same populations we looked at previously, right? Quite distinct. The new addition here is this group that we called unsubtyped. And it turned out that this is largely sort of asymptomatic, low bone marrow involvement, people with smoldering disease primarily. And that made us start to think that maybe what we were looking at is some kind of evolution in Waldenstrom's, right? Going from unsubtyped to one of these two subtypes. And this led us to use more fancy math because, why not? And again, we don't need to worry about this too much, but the goal of this kind of analysis, and it's called diffusion mapping, is to try to find like a surface or tracks, if you will, that the data points like to fall on, and then sort of line everybody up on that, right? It doesn't always work, but when it works out really beautifully. And this is what we got. And when you get something like this, you can perform an additional analysis called pseudotime, and that's saying like, hey, look, I think that this is actually something that's evolving over time, and that what we're really capturing is cells starting at one end and going to the next end. And can we sort of come up with a number that will allow us to place each sample sort of on that path, somewhere on that path from beginning to end? And so what did we see? Well, in the beginning, we see a lot of small dots. The size of the dots correspond to how much bone marrow involvement there was. And while it's hard to see, actually a ton of blue dots, right? And everyone's closely packed together. As time goes on, we see them start to separate out. And what do they separate out into? The two subtypes we just identified earlier, this B cell-like and plasma cell-like groups, right? We also see the dots get bigger, right? And while I'm not showing it here, we also see more symptomatic disease start to occur. In fact, when we look at the sort of early versus late split, you're really looking at a clinical description of progressing symptomatic disease. Now, this is another slide where I don't want you to pay too much attention to what's here. I want to actually focus on some bigger points, which is that the gene sets that we're using for these analyses are not the same gene sets we use to discover the subtypes. We can actually use these two basic concepts to really explain a lot of the differences in gene expression that we see in Waldenstrom's. And what are these two concepts? Well, we see these green dots, these are the unsubtyped, move into one of the two subtypes over time, the red and the blue. We see them move from this early evolutionary score, this early pseudotime, into this more evolved symptomatic disease. Those are the small dots going to the larger dots, right? So we can kind of superimpose these two ideas with completely separate independent gene sets to describe the evolution of Waldenstrom's. And in fact, this corresponds to a lot of what we see when we look at DNA mutations. There's differences there. When we look at clinical symptoms, also differences corresponding to these different states. So this is giving us, for the first time, a way of explaining what we've been observing all along. And again, don't worry too much about what these actual differences are. At this stage, it's not terribly important, other than just to show you that they are there. So why am I convinced that this really is disease progression? Because I think that that's something that is worthy of revisiting, because that's a big claim. And we can divide up these samples, these sort of into natural chunks, natural breaks in the data. You can see here by different colors. And then look at things like, what's the average number of mutations per tumor sample? And what we can see is that they're growing with increasing pseudotime. Right? The longer you have WM, the more mutations those cells are going to accumulate. Right? What else do you expect to see? Right? You expect more bone marrow involvement as the cells grow and expand. We see that too. The other thing that we see is we can predict, it's very predictive of how long it's going to take for someone to have need to have treatment. So time to first therapy. Now, again, this is another kind of graph that I suspect most people aren't used to seeing. It's called a Kaplan-Meier curve. And what it means is that everyone starts at 100%. That means nobody's been treated. And as you follow this time here in months, this is a proportion in each group that is now needed to have, it still remains untreated in this case. And you can see there's big differences. That curve, those two curves are very separated. And this is telling us that we can use this sort of pseudotime or evolutionary score to predict who's going to need therapy and who's not. Now, why do we care? I mean, it's a cool parlor trick, right? But why do we care? We care because if we can understand the biology, we can try to interfere with this process, right? We can develop new therapies targeting this process. We can try to understand what's driving it, what is the biology, what can we do to undermine it, to block Waldenstrom's from progressing, to try to get rid of some of these early clones, and develop new treatments, right? And that's the goal. That's why we do this. Now, the one last thing that we haven't talked about, and I realize we're running out of time here, is what does this mean? And how does this tie into what we talked about earlier? That's a good question. So I'm going to give you two examples. One is looking at the actual bone marrows of patients with Waldenstrom's. So we can look at the bone marrows of patients with Waldenstrom's, where we got these samples from, and we can say, look, when we look at these early ones, there's less WM cells, which makes sense, but there's a ton of these pink cells here. These pink cells are myeloid cells. And when you get advanced symptomatic disease, they go away and are replaced by like rings of mast cells and lots of tumor. Now, why might this happen? Well, it turns out when you look at these samples and break it down with this evolutionary score, in the beginning, they're secreting a lot of something called chemokines. And if you remember CXCR4, I talked about trying to follow chemical trails. These are the chemical trails that they're learning how to follow. And what do? They recruit myeloid cells. Right? So we see this myeloid attractants coming out really, really strong early on, and a lot of them clustering around the WM cells early on. And as that fades away with time, they go away. But that's not the interesting thing. As a matter of fact, it's the wrong question. The really interesting thing is that B cells are not supposed to know how to do this. Right? And that gets us to the real story. Right? So what is this score that we've been talking about? And how does this tie into all this crazy epigenomic stuff, cell of origin? Why did I do all this? Well, when we look at this, when we look at the pseudotime, we look at what genes are associated with it, and we look at WM cells, we can start to think about where these genes fall in normal cellular development in the blood. Oh, sorry, wrong way. So this is where WM cells are. They're stuck somewhere between B cells and plasma cells. And as we talked about, we think there might be two different subtypes, and they may have two slightly different cells of origin representing slightly different versions of where they're stuck. Cool. All right. But what about these genes I'm showing over here? Again, the names aren't super important, right? Well, CD4, that doesn't belong there. That's a T cell gene. RAG1, IGL1, well, that's a transiently expressed thing that happens, a little bit before you create a B lymphocyte. What about CD33? That's a myeloid gene, completely different branch. Right? CD34, stem cells. Right? What we're observing here is this epigenomic reprogramming of these initially mature cells relearning how to do things that stem cells and myeloid cells and T cells know how to do to help themselves grow and survive. And that's really what we're measuring. So now what we want to do, and the focus of our research, is how do we interfere with this? How can we target that process and cut it off? Why is it so important that this happens? And that's the current state of Waldenstrom's. And it's something that we're very, very excited about. Now, I'm not the only person doing this. There's a great group at the Mayo Clinic with Steve Ansel, as well as Patrizia Mondello, who is also working on different signature patterns present in Waldenstrom's and how they interact with each other. And it's really brilliant work. And one of the exciting things is that we've actually decided to try to work together. We're going to be pooling our resources, comparing our models, trying to integrate this data to get a better understanding of Waldenstrom's, and again, hopefully come up with new and better therapies for patients. And with that, I'd like to finish, and I'd like to thank you all, both for being here, but also because these studies are made possible by people like yourselves. Like, these are patients who donated their samples to make this possible. The study was funded by the IWMF, by the WMFC Canada, by Leukemia and Lymphoma Society, among others. But this is work that was done for you and funded by you. And so thank you, and I'd be happy to take any questions.
Do telomeres have any expression with Waldenstrom's?
Oh, what a great question. So telomeres are, we talked about these chromosomes, right? These 23 long strands. So, putting our scientist brains back on again, let's think about, there's some weird stuff that happens at the beginning and the end of things, right? Let's say you're trying to copy stuff, right? What do you do at the very, very end? That's always a problem. Handling that's always tricky. And so there's a special system called telomeres at the ends that help handle this process. But they also handle something else too, which is they can act as a timer for how many cellular divisions have happened. And so they can shorten over time, and it can sort of serve as a way of being like, hey, you know what? I think you've divided too many times. I think that maybe you're starting to go bad. So you know what? Instead of risking cancer, let's just call it a day, right? So cells can choose to maintain their telomeres or let them shrink over time. Now, how this plays out in WM, unfortunately, I don't know. It's a great question, though, and it was something I would love to study, but it's not something we've looked at here.
So when I have a bone marrow biopsy, my physician will use flow cytometry and characterize cells as being B cell-like or plasma cell-like.
Yes.
So it's not the same sophisticated genetic analysis that you have, but is it roughly similar? And should I expect B cell-like to mature into plasma cell-like, or is this a sort of fixed phenotype for my disease?
That's an excellent, excellent question. So one of the validations that we've done with this, for this work, is actually to validate some of these findings using flow cytometry. And the person who's been leading that effort is actually here in this room, Maria Luisa Guerrera. Can stand up and wave. A Kyle Award winner, by the way, for the IWMF. She's amazing. And so what I would, so these are different things. So B cells, normal B cells and normal plasma cells are different things. Normally what happens is you have a B cell, and when it gets stimulated, it sees it's part of your adaptive immune system, the sort of smart version of your immune system. So if a B cell, it knows how to recognize something. It doesn't know what it is, but it knows it's not part of you. There's been this extensive screening process, and we said, we don't know what you bind to, but it's not you. Cool, right? That means if it binds to anything, it's not you, it shouldn't be there. And so that will cause that B cell to grow and expand, and it'll also cause it to turn into a plasma cell, which will produce, you hear about antibodies like IgM, they produce a ton of antibody. And these antibodies flood into your system and start binding up and destroying all these bacteria and viruses. It's a great system, right? So the plasma cells are the sort of professional antibody producers, and the B cells are sort of the earlier versions that are kind of going around looking to see if they can bind to anything. Now, what we're talking about here with B cell and plasma cell-like are actually two different variants of Waldenstrom's, neither of which are really truly B cells or really truly plasma cells, somewhere in between. And one thing to sort of remember is is a cancer cell. They've kind of already gone off the rails. So, B cells and plasma cells aren't supposed to express T cell genes. Some of these do, right? Some of the genes that you'll hear other people talk about, like HCK, you'll probably hear about in Steve's talk later, is actually an earlier B cell gene. It really doesn't belong in a, in the stage where we see WM cells, but it's there. It's there, right? So they've already kind of evolved away from that system, but they do have sort of, as a basic shape or function, we do see some which are closer to what we might call normal plasma cells or closer to normal B cells, but sort of an independent thing from what we're talking about. But it's a great question.
Hello, Dr. Hunter. It's
Great
nice
to see you again.
I have a question. We're seeing all these downstream events that leading to the Waldenstrom cell.
Yeah.
What are the, where are we as far as identifying those epigenetic drivers? I know that's a loaded question, and I apologize, but I'm really interested. And congratulations to your parents as well for having raised such a fine son.
Yeah, so the short version, of course, is we don't really know yet. We're working on this. We did present at ASH last year our initial data, because one of the things we can do when you look at these epigenomic changes, a lot of them were actually directly associated with the genes I talked about. So we can go back and say, okay, like, what opened up, right? And we can look at the DNA patterns that opened up and be like, okay, cool, I don't know what that does. AT, CCG, right, beyond me, right? But what we can do is we can look for patterns. And in those patterns, the kind of things we can do is, we know DNA binding proteins. They bind to particular patterns. And then we can ask, are there enrichments for particular DNA binding patterns in these regions that are suddenly becoming open or suddenly becoming closed? And the answer is yes, very strongly yes. Now, which ones are the most important, which comes before what, and that's the stuff we don't really understand yet, but we're starting to learn who the players are. And so that's sort of the current state and the sort of level of excitement, because before we really didn't know, and whereas now we can start to really focus in on, again, what we think some of these driver events might be.
Thank you so much. This was just incredible. I think what we've just seen is not only the evolution of Waldenstroms, but we've seen the evolution of the Waldenstrom's research team. And it's just, my mind is just blown at what you have accomplished over 20 years and what you're going to accomplish over the next 30, 40 years. I feel as if we are really on the cusp of coming to some tremendous understanding. I just can't thank you enough, and your parents. So thank you very much.
Thanks again for having me.